Hi everyone, welcome back to my channel. This is your Professor Hari Krishna, and in this session we shall learn about one of the most important concepts in the field of biochemistry, that is bioenergetics. We know that in a living system all the cells will always perform work constantly and continuously in order to stay alive, to grow, and to reproduce. And in order to perform all these cellular activities, cells are in requirement of energy. So from where the energy will come to the cells to perform the cellular activities? Cells will derive the energy from the sun. But it is not always possible to expose to the sun so as to get the energy. And sun is also not available for all the 24 hours of the day. So apart from sun, cells will derive energy from nutrient molecules in order to perform the cellular activities. That is carbohydrates, lipids, as well as the protein molecules. So by using the nutrient molecules, the cells will perform the biological work such as synthesis of cellular components, generating the concentration on the electrical gradients, muscle contraction, as well as most importantly the transport of the metabolites across the membrane. In order to perform all these functions, cells will utilize the energy which can be derived from sun or else from nutrient molecules. So the bioenergetics will give a complete information about the quantitative study of the energy transformation that occurs in a living cells, as well as the nature as well as the function of the chemical processes underlying the energy transformations that occurs in the cell. So bioenergetics will help us in understanding the energy transformations that occurs in the cells, as well as the chemical reactions that are taking place, that are responsible for the energy transformations that occurs in the cell. Regarding all these things, bioenergetics will give complete information. So in a strictly speaking way, bioenergetics will give information about the energy transformations that occurs in the cell. In a strictly speaking way, in living cells, as well as the chemical processes underlying the energy transformations. That occurs in living cells. So about these things, bioenergetics will give complete information. And here we said in the cells there occurs energy transformations. So when you come to the case of the term energy, we should go with the laws of thermodynamics, and whatever the energy transformations that occurs in a cell, they are governing by the first two laws of the thermodynamics, that is the first law of thermodynamics as well as second law of thermodynamics. And according to first law of thermodynamics, we know that energy can neither be created nor be destroyed, but it can be converted from one form to another form, that what we also called as law of conservation of energy. So the living cells are having capability to transform one form of energy into another. So the first law of thermodynamics will be satisfying. So for example, cells will convert chemical energy into some other chemical components or the nutrient components, and nutrient components will be converted into chemical energy. So these transformations will be observed in case of living cells, so wherein we can notice transformation of energy from one form to another form, so that in turn the first law of thermodynamics is playing its role. And according to the second law of thermodynamics, we know that the entropy of the universe, this is according to the second law of thermodynamics, entropy of universe will always increases. So we know that if the entropy of the universe is continuously increasing, that may be either in a physical process or even it may be a chemical process also. So whatever it may be. So if in case of physical or in case of chemical processes, if the entropy of the universe is increasing, then we used to say the process is spontaneous process, right? So the second law of thermodynamics will account for the concept of entropy as well as the spontaneity of the process. So this is according to the second law of thermodynamics. So according to which entropy of the universe continuously increases in both physical and chemical processes, then that indicates the process is spontaneous, right? And the living organisms will receive the energy from their surroundings and they form the nutrient molecules or from the sunlight, and in turn they return the energy to the surroundings in equal amount, either in the form of heat or in the form of entropy, so that both first and second law are satisfying. But we said in bioenergetics, our main intention is to understand the energy transformations that are taking place in cells. So we said living cells are having capability to transform one form of energy into another form. So the energy transformation will actually occur in 3 stages. So that we shall learn under the heading of stages of energy transformation. So under energy transformation, under the stages of energy transformation, there are 3 stages. The first stage is called as photosynthesis. This is the first stage of the energy transformation, that is photosynthesis. And number 2, it is respiration that occurs in all the living cells. First one is photosynthesis, second one is respiration, and thirdly, it is the most important one, that is utilization of energy. So here we majorly observe the complete utilization of energy. So these are the 3 stages of the energy transformation. The first one is photosynthesis, second one is respiration, and third one is utilization of energy. And now we shall see the exact way of energy transformation in each and every step. So firstly, we shall consider photosynthesis. So in photosynthesis, when the green plants that consisting of photosynthetic cells, when these green plants are exposed to the solar light or solar energy. So when they are exposed to the solar light or to sunlight, they capture the solar energy, and that solar energy will be get transformed into chemical energy. Okay, so the green plants will absorb the solar energy and that converted into the chemical energy, and this chemical energy will be received by the plants, which is produced in the plants. The plants will utilize this chemical energy in order to synthesize the nutrient molecules such as carbohydrates or lipids or proteins. These nutrient molecules will be prepared by the combination of carbon dioxide and water. So these things will be collectively called as nutrient molecules. Nutrient molecules. Okay, so the solar energy will be absorbed by the green plants, that is the chlorophyll, that is the photosynthetic cells, and that solar energy will be converted into chemical energy. And by utilizing this chemical energy, plants by combination of carbon dioxide and H2O will produce carbohydrate molecules, lipids and proteins. All the materials will act as energy sources. This is the thing that actually happens in case of photosynthesis. And whatever the nutrient molecules that are synthesized in the step of photosynthesis, which is the stage 1, these nutrient molecules will be changed into another form in the second stage, that is respiration. So now we shall see what actually happens in case of respiration. So in the stage of respiration, whatever the nutrient molecules that are produced in the process of photosynthesis, that is the nutrient molecules that are produced in the stage of respiration, will be used by the animals, and these nutrient molecules will be undergoing the process of oxidation when they are consumed by the animals, and through oxidation the nutrient molecules will produce once again chemical energy through the process of respiration, which is simply nothing but oxidation. So the chemical energy was converted in the solar energy was converted into chemical energy, and that chemical energy will be converted into the form of carbohydrates, lipids and proteins, that is to the form of nutrient molecules in case of photosynthesis. So the energy which is present in the carbohydrates, lipids and proteins, because we know that these materials are actually called as the sources of energy, that is the nutrient molecules. These nutrient molecules in the stage of respiration will undergo oxidation so as to produce once again chemical energy, and whatever the chemical energy which is produced in the cells through the respiration, that will be utilized by the cells once again in order to perform the cellular activities. That is, the chemical energy which is produced in the respiration stage will be utilized by the cells in order to perform, that is, the cells will utilize the chemical energy in order to perform the cellular activities such as the synthesis of the cellular components, transportation of the metabolites across the membranes, muscle contraction, all these things will be performed by the cells. So after performing the cellular activities by the cells, whatever the amount of energy, we can't say that the whole and sole amount of chemical energy which is produced in the stage of respiration may come for use, that is, may come for the performance of work by the cells. So some amount of energy may remain unavailable, and that energy will be vomited to the surroundings in the form of heat into the surroundings. So this is the utilization of energy by the cells. So in the first stage, the solar energy will be converted into the form of chemical energy by the green photosynthetic cells, and that chemical energy will be converted into nutrient molecules, and in stage 2 the nutrient molecules will be undergoing oxidation so as to produce chemical energy. In stage 3, cells will utilize the chemical energy in order to perform the cellular activities, and after performing cellular activities, whatever the amount of energy which is unavailable to do the work by the cells, and that energy will be exerted to the surroundings in the form of heat. So these are the 3 stages of energy transformation that actually occurs in cells. In the discussion of last minute, we said the chemical energy, the total amount of chemical energy which is produced in the second stage, that is in the stage of respiration, cannot be used by the cells completely in order to perform the cellular activities. Some part of it will be emitted into the surroundings in the form of heat. One question comes to our mind: why can't cells can use the whole and sole amount of chemical energy which is generated in the respiration stage in order to perform the cellular activities? In order to answer this question, let us consider one simple example. If you consider a man-made machine wherein the energy can be converted into other forms, it means which can be used as a tool in order to convert the one form of energy into another form. So here, if you consider this man-made machine, and here in this machine, the heat energy will be transformed into other forms of energy, other useful forms of energy. That means the heat energy will be converted into mechanical energy or something else. Heat energy will be converted into other useful forms. So how these things will happen in a machine? The simple thing is, in order to convert heat energy into other useful forms, heat will allow to transform from the region of higher temperature to the region of lower temperature. Just by transforming heat energy from the region of higher temperature to the lower temperature in a machine, heat will be converted into other useful forms of energies. But whatever the way of transformation of heat energy into other useful forms, by transforming it from the region of higher temperature to the lower temperature, this thing is not possible in case of biological systems. The reason is, In biological systems, or else the biological systems are essentially isothermal in nature. That means all the biological systems are preferably, essentially, they are isothermal, means the temperature remains the constant. So if the temperature remains constant means pressure remains constant according to Gay-Lussac's law. So say for example, if you consider a human body, all the cells used to perform at a constant temperature of 37 degrees Celsius, that is normal body temperature, right? So in case of biological system, no two positions of a cell can have two different temperatures, or no two cells in a tissue can have two different temperatures. So heat energy cannot be used directly in order to carry out the cellular activities by the living cells, because there is no variation in the temperatures at any two parts of the cells or any two cells of the tissue. So heat energy cannot be used. First, the solar energy has to be converted into chemical energy, that into nutrient molecules, and once we consume the nutrient molecules by performing the oxidation in the respiration, that nutrient molecule will give the chemical energy, and that chemical energy can be used to perform the cellular activities. And now if we go back to the actual question of ours, that is, why can't we use the whole and soul amount of energy which is generated in the respiration stage to perform the cellular activities? That means whatever the energy which is produced in the respiration stage is not available to do the work by the cells. So some part of energy will be utilized to conduct or will be used to create some other things in the body, and that is not available to do the work. So whatever the amount of energy or the form of energy which is useful or which is available by the cells, provided at a constant temperature and pressure, means the whole and soul amount of energy, chemical energy, cannot be used by the cells at constant temperature and pressure to perform the cellular activities. Only a part of energy can be utilized to perform the cellular activities, so that cells cannot use the whole and soul amount of chemical energy. So only the some form of energy can be used by the cells. In a strictly speaking way, the cells will utilize the energy which is available to do the work, and the energy utilizing by the cells to perform the cellular activities, strictly speaking, the energy which is available for the cells to perform the cellular activities by the cells is simply called as free energy. That means the amount of energy which is available to do the useful work will be called as free energy. So that we can say plants and animals will be considered as the chemical engines which work under isothermal condition, that is at constant temperature and constant pressure. So here I said the word free energy. The free energy which is utilizing by the cells at constant temperature and pressure is actually called as Gibbs free energy. Gibbs free energy, which is designated with the letter capital G. And the free energy measurement is not that much easier. Even though we are measuring the things practically or experimentally, it is not that much easier to measure the Gibbs free energy directly, but we can measure the change in free energy, that is delta G. So if you consider a simple reaction which is reversible in nature, so A is in equilibrium with B, or A is reversibly changing with B. So for this particular reaction, the value of delta G can be simply calculated as this gives the difference in free energy of the product, that is B, and free energy of the reactant, that is A. So GB minus GA will give the value of delta G, that is change in free energy. This change in free energy, that is delta G, is a very important tool. It is having a great importance in the biological system, and the parameter delta G, that is change in free energy, in a strictly speaking way, change in Gibbs free energy, will be related to three more other thermodynamic properties, that is enthalpy and entropy of the system, as delta G is equals to delta H minus T delta S. So this is the relation through which we can relate free energy with enthalpy and entropy, as well as the absolute temperature. So here delta G is the change in free energy, delta H is the change in enthalpy, delta S is change in entropy, and T is the absolute temperature. That means we are expressing the temperature in terms of Kelvin. So if you consider a chemical reaction, we know the exact meanings of these three parameters. That is, delta G is called as available energy to do the useful work. H is the enthalpy, that is total heat content of the system. S is entropy, which gives the information about randomness in the system. And T is the absolute temperature. So if you consider a chemical reaction, if the products are in more disordered state than the reactants, that is, in B, if the molecules or the constituent particles are in more disordered state compared to reactants, then we used to say the entropy, that is delta S of the system, is increasing, right? So in that case, we will give delta S is equals to positive. And if you notice T delta S, so the change in entropy of the system is multiplying with absolute temperature, because the entropy change is directly related to the temperature of the system. So the term T delta S is actually represents, or else let me tell you in this way. So here it is delta H, that is total heat content of the system. So that means how much of amount of energy which is giving for the system. So we said whatever the energy which is given for the system, the whole energy is not available to do the work. Only a part of it will be available to do the work, and remaining part will be not available, and that will be exerted to the surroundings. So here the term TDS, the product of absolute temperature and change in entropy, this will give information about the portion of the change in enthalpy. So enthalpy, that is delta H, the portion of the delta H which is dissipated, which is unavailable for the useful work. That is the information we can get from the product of T into delta S. That means we said delta G is change in free energy, that gives the amount of energy available to do the useful work. So total amount of energy we are providing is delta S, and part of energy which is not available to do the work is T delta S. When you have taken the difference between the total energy given and the part of energy which is not available, we are going to get the part of energy which is available to do the useful work, that is the value of delta G. So delta G is very much important in the field of thermodynamics, which is applying in biological system. So delta G, by knowing the value of delta G, it is possible to predict the direction of the chemical reaction. That means whether the reaction is going towards the forward direction, or whether it is going towards the backward direction, or whether it is attaining the equilibrium position. This information we can get from the value of delta G. As well, by knowing the value of delta G, it is possible to evaluate the amount of useful work that can be extracted at constant temperature and pressure. And I said the delta G values are much useful in determining whether a reaction is proceeding in forward direction or backward direction. That means whether the reaction is spontaneous or non-spontaneous. This thing also we can evaluate by knowing the value of delta G. In general, for a reaction, let us consider A is changing into B in reversible way. For this reaction, or in a general way, for a particular reaction, delta G can take three values. That is, delta G may have positive value, delta G may have 0, and delta G may have negative value also. So if delta G is having positive value, that gives one information. Similarly, for delta G is equals to 0 and delta G is equals to negative. And now we shall see what is the significance of these three things, that is delta G is equals to positive, delta G is equals to 0 and delta G is equals to negative. Firstly, if delta G is equals to negative. So if delta G is equals to negative, that indicates the products are having more or a lower free energy compared to the reactants. So products are having more lower, or else you can directly say the products are having lower free energy. The products are having lower free energy than reactants. So when the energy of the products is less compared to that of reactants, then there is no possibility of conversion of products back into reactants. So the only possibility is reactants can convert into products. So the reaction will be favored in forward direction. So and that is the case, then the reaction is thermodynamically more favorable. So that means when the reactants are directly converting into products, A is giving rise to B. This is the possibility when delta G is negative, because the products will be having lower free energy compared to the reactants. So reactants are having more energy compared to products. So reactants will convert into products, and since products are having less energy, so they are unable to convert back into reactants once again. So that is a thermodynamically more favorable reactions. And since the reactants are having more energy, more free energy compared to products, so energy will liberate during the reaction, and thus these kind of reactions are called exergonic reactions. Exergonic reactions, that mean any biological reactions in which the free energy of the products are less than that of reactants. So that means the delta G of the reaction is negative, and if delta G is equals to negative, the reaction is thermodynamically more favorable. That indicates the reaction can take place on its own. That means the reaction is spontaneous. Such a kind of reactions are called exergonic reactions. Simply nothing but they are exothermal processes. And for this kind of exergonic reactions, we shall see the energy profile diagram and how it useful in explaining the energy factors. In the graph, you can see the reactant molecules are having higher energy compared to that of product molecules, so that the reactants are passing through a transition state and converts into products. I mean the reaction is favoring in forward direction, so that the product molecules are having relatively lower free energy, that is delta G is less than 0. Therefore the reaction is more favored thermodynamically and the reaction is spontaneous, so that we are observing delta G value as negative. Secondly, when delta G is positive. So if any reaction is having a positive delta G, that is change in free energy is positive, that indicates the products are having more free energy or greater free energy compared to reactants. So compared to reactants, the products will be having more free energy. So when products are having more free energy, then that indicates the forward reaction is not that much favored. Instead, the backward reaction is most favored. Means the reaction is not allowed a reaction. That means the reaction is thermodynamically unfavorable reaction, because the product formation is not taking place, since the products are having more energy compared to that of reactants. So the energy will always flow from higher level to the lower level. So products will try to convert into reactants compared to conversion of reactants into products. So in this case, conversion of A to B is not possible thermodynamically, and such kind of reactions are unfavorable. But if you want to make the reaction to take place, then we are supposed to provide the energy to the reactants to make the energy of reactants more than that of the products in order to make the reaction to proceed to produce products. That means the reaction is not taking place on its own. That means the reaction is non-spontaneous. And such kind of reactions in which the reaction is thermodynamically unfavorable and the reaction is non-spontaneous and which can takes place by the absorption of energy by the reactants, such kind of reactions are called endergonic reactions. Endergonic reaction, that means the biological reaction for which the free energy of the products is more compared to that of the reactants, and for which the value of free energy change is positive, are called as endergonic reactions. And now we shall see the energy profile diagram for endergonic reactions. In the graph, you can see the product molecules are having relatively higher free energy compared to that of reactant molecules. Therefore, if the reactants has to convert into product, they cannot take place on their own. They need more energy to convert into transition state. Again, they convert into product. But since the reaction is more favored in backward direction, that is conversion of product into reactant, because the delta G naught value is greater than 0, that is a positive value that accounts for thermodynamically unfavorable reaction, since the reaction is occurring non-spontaneously and it is required the energy supplying from outside so as to take place to produce the product molecules. Lastly, the change in free energy, that is delta G is equals to 0. If the reaction is having delta G is equals to 0, then we used to say the reaction which is reversible with B will be in the state of equilibrium. So if delta G is equals to negative, the reaction is spontaneous. If the delta G value is positive, the reaction is non-spontaneous. And if the value of delta G is equals to 0, if delta G is negative, the reaction is more favored in a forward direction. If the delta G is positive, the reaction is more favored in backward direction. If delta G is equals to 0, the reaction will favor both forward as well as backward direction, provided both forward and backward reactions will take place in the same rate, and thus the system will attain the state of equilibrium. So, by the values of delta G, that is change in standard free energy, it is possible to judge whether the reaction proceeds to completion or not, or whether the reaction is attaining the state of equilibrium or not. These things can be analyzed by looking over to the value of delta G. So if you consider a reaction A gives rise to B, which is in a reversible condition, provided maintaining the conditions of constant temperature and pressure. That is, for a biological systems, we are maintaining the isothermal conditions. And for a general reaction, say A gives rise to B at isothermal condition, that is maintaining constant temperature and pressure, the free energy equation will be given as delta G is equals to delta G naught plus RT ln concentration of product to concentration of reactant, right? So this is the generalized free energy equation, wherein delta G is the free energy change, delta G naught is the standard free energy change, R is universal gas constant, T is absolute temperature, B is concentration of product, and A is the concentration of reactant. So we said that if you consider, and here it is delta G naught, which is the free energy change under standard conditions. So what are the standard conditions? Maintaining a temperature of 298 Kelvin and maintaining the concentration of all the constituents at exactly, that is maintaining all constituent, that is concentration of product as well as concentration of product exactly equals to 1 mole per dm cube. These are the conditions for the value of delta G naught. So if you have taken this particular condition, that is both concentration of B and concentration of A is equals to 1, so ln of 1 will come and ln of 1 will be 0. So in that case, you will be getting delta G is equals to delta G naught plus the whole term will become 0. But if you consider the state when the reaction is attaining the condition of equilibrium. So at the condition of equilibrium for a reaction, strictly speaking in a biological reaction or the chemical reaction which is taking place in a biological system, at equilibrium the value of delta G, that is the free energy change, is equals to 0. And if you apply this condition to this particular equation, it will become delta G is equals to 0, delta G naught plus RT ln concentration of B to concentration of A. Or else delta G naught is equals to, plus will come to this side, it will become minus RT ln, and the ratio of concentration of product to the concentration of reactant will be taken as Q, which is nothing but reaction quotient. Or else when the concentrations are different, we will be taken as reaction quotient. But since we are applying the condition of equilibrium, the reaction quotient will be replaced with a constant KEQ, which is called as equilibrium constant. So the equation will become delta G naught is equals to minus RT ln KEQ. This is the relationship between the standard free energy change as well as the equilibrium constant. So by knowing the values of equilibrium constant of a reaction, it is possible to calculate the value of delta G naught, and thereby it is possible to predict whether the reaction is taking place in a forward direction, backward direction, or in equilibrium state. So for example, if the value of KEQ, that is the equilibrium constant, is exactly equals to 1. So if K is Q is equals to 1, ln of 1 will become 0. Therefore, the whole term will become 0. Therefore, delta G naught is equals to 0. That means the reaction is at equilibrium. And if the value of equilibrium constant, that is KEQ, is greater than 1, so if it is having a positive value, then the value of delta G naught will be negative. Then the reaction will proceed in a forward direction. And if the value of KEQ, that is the equilibrium constant, is less than 1, means the KEQ will be having a negative value. So minus is already here. So here also if you're getting minus, means minus into minus you will be getting plus. So delta G value will be positive. Then the reaction is more favored to occur in backward direction. So this is thermodynamically favored. This is thermodynamically not favored reaction. So in this way, by relating the value of standard free energy change as well as equilibrium constant for the chemical reactions that occurs in biological system, we can judge whether a particular cellular activity or cellular reaction is spontaneous, non-spontaneous, thermodynamically favorable or not. All these things can be observed. We said that under standard condition, say at a temperature of 298 Kelvin and under a pressure of 1 atmosphere pressure and having a concentrations of 1 mole per dm cube as well as at a pH of 7. At a pH of 7, if you notice the sign of the standard free energy change, that is delta G naught, we can judge whether the reaction can occur spontaneously or non-spontaneously. As well, we can say whether the reaction is present in equilibrium state or not. So if you consider the hydrolysis of glucose 6-phosphate. So if you consider the hydrolysis of this particular material, hydrolysis. So upon hydrolysis, glucose 6-phosphate, you will be getting glucose with the regeneration of inorganic phosphate. For this reaction, the value of delta G naught will comes equals to minus 13.8 kilojoules per mole. So here we are having delta G naught with the negative sign. So this is a spontaneous process. So if you consider one more thing, that is glycyl glycine, that is a peptide. So if you subject this dipeptide for the process of hydrolysis, glycyl glycine, so you will be getting two molecules of glycine. So the dipeptide has been converted into amino acid, for which the value of delta G naught will be given as minus 9.2 kilojoules per mole. You can notice for this reaction, delta G naught value is minus 13.8 kilojoules per mole, and for this it is minus 9.2 kilojoules per mole. So this is more negative compared to this one. So the hydrolysis of glucose 6-phosphate to give glucose is thermodynamically more favorable compared to the breaking of a dipeptide to produce simple amino acid molecule. So these things can be said by looking over into the values of delta G naught values. And if you consider one of the most important reaction that occurs in biological system, that is oxidation of glucose in respiration. So when glucose is bonded with oxygen, it's going to produce 6 moles of carbon dioxide and 6 moles of water. For this reaction, the value of delta G naught is equals to minus 2840 kilojoules per mole. So this is having the very greater negative delta G naught value, and for this reason, compared to these two, this is the most thermodynamically favorable reaction that actually happens at each breath of our respiration, and because of this we are getting more energy, and that released energy will be utilized by the cells to make the body, to maintain the body temperature, to perform the cellular activities and everything. And this the whole and soul thing about the concept of bioenergetics in the case of the topic biological oxidation. And hope the video is useful for you people. If it is useful, if you felt it is useful, please like it, share it, and please do subscribe to my channel and press the bell icon so that you will get the notification for any newer update in my channel. And in our next video, we shall learn about the energy coupling as well as the energy rich component in the biological system. Thank you very much.